Hey everyone, how's it going? This is the second part of our two-part video series on the histamine signaling pathway. In the first video, we looked at the production, packaging, and release steps of histamine. And today we're going to look at the response and termination steps. So we're going to look at how histamine binds onto different receptors to produce various responses, and then how these responses eventually get terminated. Let's go. So after histamine gets released from various cells such as neurons and mast cells, it leaves the cell and it goes into an area like your synapse or like your extracellular space, and then it can go over and bind to different receptors found on various different tissues. Now, the thing is, there's a lots of different tissues that have lots of different receptors to which histamine can bind onto. And different tissues are found in different areas of the body. So depending on what area of the body you're looking at, you'll have different tissues with different available receptors. So histamine can go and bind onto them and produce a variety of different responses. So histamine responses in one part of the body might be different than in another part of the body. Now there are four main receptors that histamine can bind to. So here we have our four different types of histamine receptors that histamine can go and bind onto to produce various different responses. And as we said before, your four histamine receptors are found on various different tissues and organs throughout your body. But before we get into these receptors and what they do and where they're found, I do want to mention that I really recommend watching my past videos that I made about G proteins and second messengers such as CAMP, DAG, and IP3. I've linked all these videos down in the description below, and it's important to understand these because all of these histamine receptors are GPCRs that are linked to various G proteins to control the production of various second messengers. So understanding how G proteins work and how GPCRs work is critical to understanding how these different receptors function. It gives a good foundational base of understanding. So I would really recommend watching those videos first, but with that, let's get into what these four receptors do. So your four different histamine receptors, they're all GPCRs, and they're conveniently named H1 receptors, H2 receptors, H3, and H4 receptors. So they're all GPCRs that are linked to different G proteins. So H1 receptors are linked to GQ proteins, which increase the production of your second messengers IP3 and DAG. And these second messengers can go off and produce various responses. Now H2 receptors, on the other hand, are linked to GS proteins, and this increases the production of your second messenger CAMP, which is another second messenger that can go and do its own responses. Now H3 and H4, histamine receptors, they are both linked to GI proteins. And GI proteins, they actually lower the production of CAMP. And this may seem counterintuitive because if you're trying to produce a response, why are you lowering the amount of a second messenger? Well, actually, lowering the amount of a second messenger, CAMP, can still have different effects and responses. So here's a table to help break down the locations and responses of your various different histamine receptors. So H1 receptors, they are found on smooth muscle cells, the brain, and endothelial cells. Remember that the endothelial cells, they form the inner layer of your vessels. And H1 receptors, they produce responses such as allergy responses like itching, and they also help keep you awake. Then you have H2 receptors. These are found on your gastric mucosa, various mast cells, and on cardiac muscle. H2 receptors, they produce the response of gastric acid secretion and help with digestion. So H3 receptors, these ones are a bit more complicated and unique. They're found on presynaptic neurons. So what I mean by that is, here you have your neuron, which releases histamine into your synapse, which is the gap between your neuron and your postsynaptic cell, such as another neuron or another tissue. So synapse in the middle, presynaptic neuron before, and postsynaptic cell afterwards. Now, most of the time, receptors will be found on postsynaptic tissues, and histamine can go and bind onto these receptors to produce responses. But H3 receptors are unique in that they are found not on your postsynaptic tissues, but instead on your presynaptic tissues. So This might seem a bit weird, but histamine gets released and then it actually goes back and binds onto receptors of the same neuron that released it. So remember, this neuron released histamine into the synapse, and then histamine can go back and bind onto receptors of the same neuron that released it, the presynaptic neuron. And when it does this, it basically activates a negative feedback loop. In order for this release process to work, calcium ions must enter the neuron via ion channels. This is important for release. And what these receptors do when they're activated by histamine is they inactivate these channels, and they do other things to prevent the release of histamine. So it's like a negative feedback loop. The more histamine you have getting released, the more histamine will come and bind onto these receptors, and the more histamine release will be prevented. And this is done to keep histamine release under control so that you don't have an excessive amount of histamine being released. So H3 receptors, they control histamine levels by lowering the release of histamine via a negative feedback loop. One thing to note is that because H3 receptors, they lower histamine release in order to keep levels under control, they're lowering the amount of available histamine there is to bind onto your other histamine receptors. And because of that, they can effectively lower some of histamine's responses. They can kind of counter it. So for example, histamine normally goes and binds onto H1 receptors to keep you awake. But because H3 receptors lower the release of histamine, they can counter this and therefore cause sleepiness. And then finally, there's H4 receptors. They are found in your white blood cells, mast cells, as well as helper T cells. And they are responsible for immune responses as well as inflammation. So there's other responses that histamine produces, but these are your main ones, produced by histamine receptors 1, 2, 3, and 4. And as you can see, these responses are greatly responsible for immune responses and allergic responses, but also for other bodily functions such as digestion. As we said, gastric acid secretion is important for digestion. Now, while all these responses are essential and important, they can be undesirable and even dangerous if they get excessive and out of control. So here in the red column, I've written down some effects that can happen if your responses get out of control. So H1 receptors, like we said, if they're excessively activated, it can lead to allergic responses, including itchiness, bumps, hives. H2 receptors, which are responsible for gastric acid secretion. So if this happens excessively, then gastric acid can damage your stomach lining and cause painful ulcers. H3 receptors, because they lower the release of histamine, if they're excessively activated, they can lower histamine release a bit too much, which can counter some of the important effects of histamine, such as keeping you awake, and this can lead to something called narcolepsy, which is when your brain's sleep-wake cycle gets messed up, so you can have sleep paralysis and sleepiness throughout the daytime. And then H4 receptors, if they're activated excessively, it can lead to excessive and potentially dangerous immune responses and inflammation. So now that we understand some of histamine's responses and negative effects that can happen if these responses get out of control, this is a perfect segue into our last, final step of the signaling pathway, which is termination. As we've just seen, responses of histamine can get dangerous or uncomfortable if they go out of control, so it's important to terminate these responses. Now, lots of pharmaceutical companies have made medicines, for example, Benadryl, which help to lower histamine's responses if they're being excessive. But your body also has innate termination mechanisms to help control histamine levels. Let's take a look at these mechanisms. So I've actually already made a video in the past fully dedicated to different termination mechanisms and how they work. And I'd really recommend watching it for an in-depth explanation on these, because as much as I'd love to, I can't go too in-depth into how these different termination mechanisms work in this video. There's just too much information, too much to pack into a video with everything else. So I really recommend watching that video for some foundational understanding, especially for this second termination mechanism, receptor desensitization. It's definitely the most complicated one. But yeah, so I'll leave all those videos linked down in the description below. So your first termination mechanism, uptake, is pretty self-explanatory. So basically, after your histamine gets released from whatever cell it was stored in, so here I've drawn a neuron, but it could also be like a mast cell or something else. Basically, after histamine gets released, if you have excessive histamine, it can be reuptaken into your cell and stored for later. So let's say some kind of stimuli happens, like an infection, and your body's going to release lots and lots of stored histamine, but then your body's like, whoa, we've released way too much histamine. We don't need all this. We don't want the response to get out of control. Let's take some back. So then some of the histamine is going to be pumped back into the cell that it came from, or maybe another cell, and then it can get restored back in your vesicle or your granule until later on when it's really needed. And that is the first termination mechanism, known as uptake. So that's the first one. And the next one is receptor desensitization. So the next termination mechanism of receptor desensitization, as I said before, it's definitely the most complicated one, so we're not going to get into it too in depth in this video. If you want to learn more about it, be sure to check out my video that I linked down below, all about termination mechanisms. But here's like the general idea. So histamine gets released, and let's say it's in your synapse or your extracellular space, and let's say you have like an excessive amount of histamine. The excessive amount of histamine is going to go and bind to a bunch of different receptors on a tissue, and that's going to produce lots and lots of response inside your cell or inside your tissue. So you have lots of histamine going on and binding onto lots of receptors to produce lots of response. Now, as we said and as we know, we don't want too much response. We want to be able to lower this and control this. So what receptor desensitization is that some of these receptors, let's say like half of them, can get desensitized or deactivated. And this is done by basically pulling these receptors into the cell so that histamine can no longer go on and bind onto them and activate them. So these receptors get pulled into the cell. They're no longer on the cell membrane. So now if you look at the available receptors that histamine has to bind onto, you only have 4 receptors instead of your 8 that you had before. So as a result, less receptors histamine can go and bind onto, less response will be produced inside the cell. Less response will be produced. And so this is a effective termination mechanism, and it's also a temporary one, because let's say after a while your histamine levels reduce, they go back down to normal, then resensitization can happen. So those histamine receptors that were pulled inside your cell, they can be returned back up to the surface, the cell surface, and you can go back to the original number of receptors that you once had. So it's a cycle of desensitization when you have too much receptors and too much response, and then resensitization when response levels go back to normal. A bit more complicated than that, there's a few other things involved, so be sure to check out that video linked in the description below, but that's the general idea of receptor desensitization. So the third termination mechanism, enzyme degradation, this is the one that we're going to focus most on in this video. As the name suggests, it's when your body's enzymes help to degrade histamine. And here are your 2 common enzymes that degrade histamine. There's DAO, which is diamine oxidase, and HNMT, which is histamine and methyltransferase. So your first termination mechanism, diamine oxidase or DAO, it turns histamine into imidazole acetaldehyde, effectively deactivating it. And this can be further turned into imidazole acetic acid. Note that the word imidazole refers to the imidazole ring structure, which is the foundation or the backbone of histamine. And then your second termination enzyme is HNMT, or histamine and methyltransferase. As the name methyltransferase suggests, it adds a methyl group to histamine, turning histamine into N-methylhistamine, effectively deactivating it. So now we know that we have 2 different enzymes which are both good at terminating and degrading histamine. But what's the point of having both if they both do the same thing? Well, actually, they both work well in different situations. So diamine oxidase is found in a few different tissue types: your small intestine, ascending colon, placenta, and kidneys. It's what's known as a secretory protein, meaning it gets secreted out of cells and goes out into the extracellular space to do its job. And because of this, it is responsible for breaking down and degrading extracellular histamine. So, for example, histamine that gets released from neurons or mast cells and is now in the extracellular space, it gets regulated and terminated by diamine oxidase. Now, because diamine oxidase breaks down extracellular histamine, It's also responsible for breaking down histamine that enters via food. So, because remember, the diamine oxidase is found in your digestive tract. So when you eat food, diamine oxidase can help to break down the histamine that's entering through the food to prevent an excess of histamine. And some examples of foods that are very rich in histamine include pickles, fish, cheese, and red wine. So diamine oxidase in your digestive tract is very important. It helps keep an important balance so that histamine that's entering via food doesn't become excessive and cause adverse effects. It keeps things under control. But this balance can be thrown off. Some drugs and medicines may inhibit diamine oxidase. So if you take these drugs and you happen to eat too much food containing too much histamine, then there won't be enough diamine oxidase to terminate the histamine, and this can lead to something called histamine intolerance, where you have excess histamine and not enough DAO to compensate. And this can lead to symptoms like itchiness, headache, dizziness, high heart rate, sneezing, nausea, and more, because the balance between incoming histamine and protective diamine oxidase is thrown off. So on the other hand, you have HNMT, histamine and methyltransferase, and it's found in a greater variety of organs. Unlike diamine oxidase, which is only found in a couple of organs, histamine and methyltransferase can be found in many more, including the kidneys, the liver, the colon, the prostate, the ovaries, the spinal cord, just to name a few. And instead of being a secretory protein like diamine oxidase is, it's cytoplasmic, meaning it rests in the cytoplasm within cells. So HNMT is either produced in your cells or it comes from outside and enters your cell. And then it waits inside your cell in the cytoplasm until it needs to get into action. And the reason it waits inside the cell is because unlike diamine oxidase, which degrades extracellular histamine, HNMT degrades intracellular histamine. So if there's excess histamine inside your cell, HNMT will be responsible for degrading that. So let's just review some key differences between our two termination enzymes, DAO and HNMT. DAO is found in a few tissue types, whereas HNMT is found in a much greater variety of tissues. DAO is a secretory protein. It leaves cells to go into the extracellular space where it degrades extracellular histamine, whereas HNMT stays in the cytoplasm of cells where it degrades intracellular histamine. And then a final difference that we didn't really mention before is that DAO, it's a bit less selective. It can also degrade other messenger molecules such as cadaverine and putrescine, whereas HNMT is a lot more selective to just histamine. But basically, your two termination enzymes, as well as with your other termination mechanisms like receptor desensitization and uptake into cells, they all work together to help keep histamine levels under control and selectively terminate histamine responses, preventing them from getting out of control. And that basically concludes our two-part video series on the histamine signaling pathway. Thank you guys so much for watching. I really hope you enjoyed and found this video useful and informative. If you have any questions, comments, or suggestions, or you would like me to make a video about something specific, please leave them down in the comment section below. I would read everything and take all suggestions into account. In the next video, we might look at the signaling pathway of a new messenger molecule, or maybe we'll switch things up and look at how orthosteric and allosteric antagonists and modulators work. I'm not 100% sure yet, but we'll see. Until then, thanks again for watching and take care.